Hello, everyone. My name is Manav Thakkar. I am from Los Gatos, California, and it's finally nice to have some sunshine after a whole while of rain. Anyway, my topic today is genetics, neurochemistry, and neuroanatomy of addiction by me, and my mentor was Dr. Rahul Patel. So let's begin with just a brief overview. So first, let's go over what is addiction, what is the genetic basis of addiction, and look at the means and methods of addiction through three different lenses, which are tobacco, alcohol, and opioids. And finally, what are techniques that are already used, but we can also apply them to addiction. The reason why I chose this is because I have a personal connection with each one of the substances which I've chosen. For example, as you can see on the screen, these are pictures of my grandfather who suffered from a tobacco addiction since he was a young man. But firstly, what is addiction? Well, addiction is broadly defined as the condition of being addicted to a particular substance, and this features distinct phases. These include initiation, experimentation, regular usage, risky usage, dependence, addiction, crisis, and finally treatment. According to the National Institutes of Drug Abuse, over an overall 12-month period of prevalence of addiction among United States adults varies from anywhere between 15% to 61%. While diffusing and far-reaching, emerging research indicates that a high inter-individual variability in susceptibility to addiction and considerable substance-specific variability. Essentially, what that means is that addiction may be mediated by a complex gene environmental factors that we have to look for in each individual substance. So let's go on to the central question of today, which is what is the genetics, neurochemistry, and neuroanatomy of addiction, and how will it impact our society today? First, let's go over some quick overview of genetics specifically. According to the Blueprint of Health and Diseases of Genetics tells us that genetics control multiple different factors. These include numbers and types of receptors in people's brains, how quickly their bodies are able to metabolize drugs, and how well they're able to respond to different medications. In addition, did you know that there are more likely to be more dopamine receptors in the offspring of a person who is addicted to a particular substance rather than the offspring of a person who's not addicted to any particular substance? In fact, general genes responsible for addiction are including, as shown on the screen, the CHRNA5, CHRNA7, CHRNB1, and CHRNB3. So first, let's go over the first substance which I mentioned before about tobacco. Specifically, when it comes to tobacco and genetics, accounts for about 75% of a person's inclination to begin smoking, according to University of Pennsylvania's psychologist Karen Lehrman. 60% of the tendency to even become addicted in the first place, and 54% of one's ability to quit are controlled by genetics. In fact, did you know that twin studies of twin groups suggest that 40 to 70% of risk factors are hereditary? Nicotine releases a chemical known as dopamine in the same regions of the brain as other addictive drugs. These cause mood-altering changes to create a person that feels temporarily good. The substance of tobacco is also the reason why I was inspired for this topic. As I'm sure I just mentioned, my grandfather was addicted to tobacco from a very young age. However, here are some quick results about smoking, which I'm sure all of you do know. They include cancer, heart disease, stroke, lung diseases, diabetes, and in fact, chronic obstructive pulmonary diseases, including chronic bronchitis. However, this also leads to increased risk of tuberculosis, certain eye disease, and problems of the immune system leading to rheumatoid arthritis. And these are more well-known results of tobacco and nicotine dependence, which include schizophrenia, depression, as shown to be on a high rise in our society today, neuroticism, self-reported risk-taking, high body mass index, which essentially means the amount of fat or the amount of fat that's within you, and also alcohol use disorders. However, what we have to remember is the amount of complexities that we have within this idea of genetics being influenced on different drugs. These include how to determine the specificity of genetic influences and clarifying the role of the interactive contributions. However, there are many ways to even address this in the first place. There are intermediate phenotypic approaches which seek to identify the intervening proximal mechanisms directly correlated with genetic risk. In fact, according to the Gene Type Tissue and Expression Project, DNA variants found in the DMT3B gene are most common in people of European and African descent. Specifically, the RS910083 C allele and a cis-expressing QTL variant have been shown to have an increased DMT3B expression within the adult cerebellum. This means that this results into a more development of nicotine dependence, heavier smoking, lung cancer, and enhanced sexuality. Current pharmaceutical treatments include the idea that the neuroadaptations may evolve to counteract the diminished glutamate and cholinergic transmission that play a key role in nicotine addiction-induced withdrawal symptoms. These include several pharmaceutical agents. Among those are acetylcholine, dopamine, glutamate, and Y-aminobutyric acid, as well as many different signaling pathways that are highly anticipated in their use of treatment for withdrawal symptoms due to nicotine dependence. Next, let's go into the next substance of alcohol. As a person who has a brother who suffers from alcoholism, I'm well worried for my nephews. However, current estimated heritability for alcohol use disorders is approximately 50 to 60% of total phenotypic diversity. Two genes that are being currently identified for alcohol metabolism are ADH1B and ALDH2. These are the strongest known effects on the possibility of alcoholism even developing in the first place. However, there are other genes that do have a possible impact. These are including GABRA2, CHRM2, KCNJ6, and AUTS2. Twin studies in the United States and Europe have been shown to have a 45% to 65% of alcoholism due to genetics. In fact, animal studies also agree with this range, as shown by mice and rats have been selectively bred for many traits, which include these, and also include alcohol preference, alcohol sensitivity, and withdrawal susceptibility. We could use genetically select these traits for indicates that there has to be some sort of genetics basis for them, and that different genes contribute to different aspects of the phenotype. This means that this also stimulated the release of dopamine and endorphins within the brain to produce feelings of pleasure and satisfaction, and act as a natural painkiller. Next, when we look at the potential short-term effects of alcohol, I'm sure you guys all do know this, but to go over one more time, they include hangover and alcohol poisoning, falls and accidents, more likely effective conflicts, lower inhibitions, and risky behaviors. However, this also depends on how much the person drinks, as other factors such as hydration and food consumption also play a factor. In fact, according to the World Health Organization, 5.3% of all deaths globally were caused because of alcohol consumption. Worldwide, more men die as a result of alcohol consumption than women. In the long term, alcohol consumption can also affect all aspects of a person's life, including the physical and mental health, work performance, and finance and relationships. Results from variations of genes and different social backgrounds also play a factor as well. Next, when we look at this as a whole, we must also analyze the neurotransmitters that play a role. These include dopamine, 5-HT, Y-aminobutyric acid, glutamate, norepinephrine, and GABA and serotonin. We also remember that glutamate also acts as an NMDA receptor. These impacts mean that findings indicate there are certain biological, genetical, and neurological abnormalities in people associated with the P300 amplitude as shown on the screen. Positive drinking expectancies include personality characteristics, and devious proneness, and factors may appear in boys and girls as they transition to adulthood differently. Specifically, boys begin to manifest a constellation of factors that place them at greater risk for disruptive drinking, including a lower response to alcohol, later maturation of brain structures and executive function, greater estimates of perceived alcohol use, and socialization into traditional gender roles. Next, let's go over the final substance which I researched, which is the opioids. As I'm sure you guys heard from previous speakers, by the late 1990s, pharmaceutical companies had ensured that the medical community that patients would not become addicted to opioid pain relievers. Yet, as we see them today, there is a huge opioid crisis, specifically in Ohio, that surfaced since 2017. According to the National Health Institute of Drug Abuse, we see that 40 to 60% of a person's susceptibility to addiction is related to genetic factors, but this also includes the influence of environmental factors to work on these genes. There's a notion that opioids do not have a clear pattern of inheritance, yet there is actually clear genetic evidence to back up this risk. In fact, with heritability estimates ranging from 23 to 54 years in twin and family studies, 16 polymorphisms within the ORPM1 gene have indicated that the expression levels are in the prefrontal cortex. Specifically, there are certain alleles that we must focus on that have been associated with opioid dependence, especially when we focus on the C allele of the RS3778150 and the RS1799971 allele. In fact, this raises the idea of genetic dependence, as different findings of the RS1799971 in the different populations are partially due to relevant genetic background differences. In fact, when we look at opioids as a whole, I'm sure you guys all know that they are highly addictive. However, there are different parts of the brain that they affect, including the prefrontal cortex, nucleus accumbens, and also the VTA. They activate powerful reward centers in the brain to trigger the release of endorphins. This muffles the perception of pain, and boosts feelings of pleasure. This also results into slow breathing, loss of motivation, cognition, pleasure, stress relief, drug consumption, decision-making, memory, and especially results into more craving. The release of inhibitors such as piperoxine are also shown to be prevalent. But yet these are some techniques that are already available to us as a whole, but yet we do not use them. For example, PET and fMRI scans, which I'll explain later how they actually are used, but we must remember this big idea. As tech advances, there appears to be a rise in studies of genetic factors relating to how they affect addiction. Essentially, the big idea is understanding these models with environmental factors that will, similar to humans, will be able to reveal the neurological pathways of stimuli for addicts. Lower level of D2 receptors results into an imbalance of reward and conditioning, as there is decrease in baseline activity in the front brain. This means that the person who's addicted can become more impulsive, potentially resulting to drug compulsiveness, abuse, and loss of control of the reward-seeking behavior. Like I mentioned before, there are two types of examples we can use: PET scanning and fMRI. How does a PET scan work? Essentially, a PET scan imaging is based on radio tracers labeled with short-lived position emitting isotopes, which can measure at a very low concentrations. Therefore, PET scans can be used to measure labeled compounds and that selectively bind to specific receptors, transporters, or enzyme types at all concentrations that do not perturb function. Including fMRI is another one, which is a measurement based on the changes in magnetic property, essentially believing that if generally believed that the activation signal generated from fMRI results into different magnetic properties of oxygenated versus deoxygenated hemoglobin, meaning that we can determine which parts of the brains are being used, as they will need the most oxygen. The big idea, which I'm trying to push towards you today, is that addiction finds its basis in complex gene environmental interactions, and the level of interactions of individual genes are based in brain circuits. When we look at medication availability today, we find that scientists are developing other medications to treat stimulants and cannabis addictions. Looking at a substance, all users become addicted to one due to feeling of pleasure that they receive once they take it. This also means that users are addicted to that feeling of dopamine that's being released in the nervous systems. The big issues which I presented today are including these. First, although there are genetic factors contributing to tobacco and alcohol problems, as well as opioids, that have been well established through adoption, twin and family studies, specific genes yet remain to be identified and their mode of action to be determined. Everyone is unique in their own means and methods of addiction. This means that there's no one common pill that could be used to stop the onset of addiction or to the magnitude in which addiction affects people, such as Tylenol for pain and so forth. However, when examining these three substances, there are some similar hopes. In fact, all three contain similar genetic risk. In addition, they all are involving similar neurochemical pathways. Dopamine hypothesis of addiction become a major pillar for addiction appeal. However, research is extremely lacking. Thank you so much for listening. Any questions?
Awesome. Thank you so much, Manav. I do have a question. Based on your research, would you expect genetics to play a role in all substance addictions?
Correct. Yes, exactly. Because most substance addictions do result into more activation of pathways, especially similar neurological pathways, as they all seem to affect the reward-seeking center of the brain, as well as they also all release similar neurochemicals, such as dopamine and serotonin, to help you feel more happy and more pleasurable when actually taking these drugs.
Gotcha. And you mentioned, I think at the beginning, that sometimes ethnicity plays a role in different gene variants. Is this also true for different types of substances?
It's shown to evolve in specifically alcohol, tobacco, and opioids. As I have done research too much on other substances, there doesn't seem to be too much genetic similarity between those three substances specifically and other substances. But yes, there's shown to be a little bit of promise in those other ones.
Gotcha. And next steps for this, do you have any other research that you would like to do?
Yes, absolutely. I want to find out if, like I said, there may be one common pill, but we can generalize pills based off of certain factors, such as type of substance that are addicted to people or types of even ethnicity. We can actually find a way to combat addiction, so that way that's not such a big problem affecting our society today.
Gotcha. Yeah. And what kind of drove you to do this type of project?
Like I mentioned before my presentation, it was my grandfather. He passed away recently due to his heart stopping because of onset tobacco addiction, since he was a very young man. So kind of he was the inspiration behind me even starting this project in the first place.
Gotcha. Sorry to hear that, but